anatomy, external anatomy anyway, and some of the liver's functions, and then we started to talk about the internal anatomy before we sort of ran out of time. So the liver is organized into these hexagonal, hexagon-shaped regions called sinusoids. So each of these hexagons shares a common pattern, and that is blood enters from the outside. So at each of the corners of this hexagon right here, we have what's called the portal or the hepatic triad, which is a branch of the portal vein, which is coming from the digestive tract, a branch of the hepatic artery, which is coming from the aorta, and then a branch of the bile duct system, which is going to carry bile that's produced by these cells out of the liver. So coming in from the outside, we have the blood. The blood flows towards the center of the hexagon, where there's a large vein called the central vein. And what we see is that the arterial blood and the blood from the portal vein, they mix together. And then what comes out of the central vein then is the blood that's been processed from the digestive tract, as well as the blood that came in through the hepatic artery to get these cells the oxygen that they need. So comes in from the outside and then goes down through the middle. All right. So and each one of these things is called a sinusoid. All right. So if we sort of zoom in and look at it, one of the things that we see is that the blood goes in one direction from corner to center. The bile goes in the opposite direction because the bile is made by each of these liver cells. It's collected by these little tiny tubes called canaliculi, and then the bile is moved to that branch of the bile duct. So while the blood is going from the outside to the middle, the bile is going from the middle to the outside. So we have this sort of passing in two different directions. And then, so bile is made by all of these liver cells. It's a very complex secretion. It has all kinds of stuff in it. For our purposes in the digestive tract and in digestion, the important stuff in bile are called bile salts, which are detergent molecules that break up fats into very little tiny pieces. And when we talk about fat metabolism, probably next time, we'll see in more detail how that is important. But for today, just know that each of these hepatocytes, which is a liver cell, produces bile. It's collected by these canaliculi, and then it arrives at the bile duct system at the corner. And as these bile ducts join together to form ever larger ducts until eventually they leave the liver at what's called the common bile duct, which we'll see here in just a minute. All right. So bile is produced all the time, but it's only needed when digestion is occurring. So in order to sort of deal with that problem, we have the gallbladder, which stores bile when it's not needed by the digestive system. So if we look at so what are we looking at here? All right, so there's the gallbladder. Here's the liver, right? And the gallbladder has a neck or a tube that connects it to the rest of the bile duct system. I'm going to turn that light back off because that's really washing out the picture. All right. Because this is important anatomy right here. Okay, so the gallbladder itself is relatively simple. It has a fundus or a dome-shaped top. It has a body, and then it has a neck where it connects. So here's the liver. Coming out of the liver at that porta hepatis, or the hilum of the liver, we have two branches of the bile duct system. So we have the right and left hepatic ducts. They join together to form the common hepatic duct. And coming out of that hepatic duct. This is the bile duct system. So the bile has been collected by all those canaliculi and branches of the bile duct, and eventually, where that exits the liver, we call it the common hepatic duct. The common hepatic duct joins with the duct that is leading to the gallbladder. We call that the cystic duct. So that's this right here. So you can see that there's kind of this Y. The hepatic duct and the cystic duct join together, and then what we call the after that, now it's the common bile duct. So hepatic duct comes from the liver, cystic duct comes from the gallbladder. When they join together, now they're the common bile duct. And the common bile duct then empties, you can't really see it very well, but it goes down underneath the stomach, underneath and through the pancreas, and then it's going to join with the duodenum right here. So the secretion from the liver and the gallbladder enter the duodenum at this little spot right here. And that spot is called the duodenal papillae. Okay? A papilla is a hill. So it's this little hill-like structure right here. And so if we zoom in on that little hill, we can see here's a common bile duct coming down. So that's this piece. And then also the duct of the pancreas, which is coming here from the side, joins with it. So you can see the common bile duct, pancreatic duct, they join together. And then together, they empty into the duodenum at that duodenal papillae. All right? And because the secretions of these two organs, the liver and the pancreas, are not needed all the time, there's a valve that controls this duct system at this point. So, and we call it the hepatopancreatic sphincter. Hepato for liver, pancreatic for pancreas, sphincter because that's what it is. It closes and opens just like a sphincter does. This used to have a different name that is in many ways more commonly used, and it's called the sphincter of Oddi, O-D-I. The sphincter of Oddi is another name for the hepatopancreatic sphincter. All right, so common bile duct, pancreatic duct, duodenal papillae. When the sphincter is open, secretions flow into the duodenum, and when the sphincter is closed, obviously they do not. All right, so that's from liver and gallbladder down into the small intestine. Well, so if the pancreatic sphincter or the hepatopancreatic sphincter is closed, and if the liver is making bile all the time, well, where does it go? So in that case, we've got bile that's flowing out of the liver, but it can't flow into the duodenum because the door is closed. So in that case, what happens is that bile that's being produced backs up into the gallbladder and fills the gallbladder. So when this sphincter is closed, the bile that's being produced, instead of going into the duodenum, it's going into the gallbladder instead. So to be stored up. And as the gallbladder fills, water is removed from the bile. So it's the bile that's in the gallbladder when the sphincter is closed gets more and more concentrated all the time. So it essentially gets stronger and stronger until you eat again, that sphincter opens, and then the gallbladder contracts, emptying itself. So the hormone cholecystokinin that we talked about last time, one of its primary functions is to get the gallbladder to contract and essentially expel its contents into the duodenum through that sphincter. All right. So why is this anatomy important? Because gallbladder problems are very, very common. The bile, as it becomes more concentrated in between meals, the bile has all kinds of stuff dissolved in it. And that stuff can precipitate. In other words, it can come out of solution and form little stones or little crystals. Well, those crystals can end up getting stuck in this duct system. So, for example, if you had a gallstone or a gall crystal that got stuck right at this spot right here, you would have a major problem because now your bile duct is blocked and your pancreatic duct is blocked. And those things do happen. So this anatomy is reasonably important because it is affected commonly by with gallbladder disease in particular. All right. Okay, so gallbladder and the duct system. All right. So, oh, I kind of already covered this. So, we all know that you can live without a gallbladder, right? Because we've all heard that people get their gallbladders removed. So in the normal case where you do have a gallbladder, here's the liver secretes bile all the time. When that sphincter is closed, instead of that bile going out into the duodenum, instead it goes back up into the gallbladder where it fills and gets more concentrated. When CCK occurs from fat in the duodenum, it triggers the gallbladder to contract, pushing that bile into the duodenum, and the sphincter relaxes to allow that to happen. All right, so what happens when you don't have a gallbladder? Well, essentially, there is no storage then, because there's no place to store it. The liver still makes bile all the time, but in the absence of a gallbladder, this sphincter is kind of always open, at least a little. So the bile is basically being made and deposited into the duodenum all the time. And it turns out that doesn't really cause any bad harm. The bile in the intestine, or the bile does no harm in the intestine, so the fact that it's present there all the time doesn't hurt anything. So some of the stuff that's in bile we want to hold on to. So there's a recycling system that's present. So in the distal part of the ileum, the third part of the small intestine, the important good stuff in bile is reabsorbed and transported back to the liver so that it can be recycled. So in the absence of a gallbladder, there's secretion and recycling that happens instead of storage and concentration, which is what happens when you have a gallbladder. All right, so then the last organ of the accessory organs is the pancreas. And it's a pretty boring looking organ. It's a giant gland, essentially. It has a head right here, a body here, kind of squarish, and then a tail that sort of points over towards the spleen. So the spleen is right about here. Some people say it looks a little bit like a fish or a tadpole. It's got a head, a body, and a tail. Running through the center of the gland is a duct called the pancreatic duct, not surprisingly. In a lot of people, 3 to 10%, your book says, the pancreatic duct branches before it enters the duodenum. But that means in 90% of people it doesn't. Okay? So in most people, there's just a single duct that empties at that duodenal papillae. So the common bile duct comes behind it, the pancreatic duct comes through the center of the pancreas, and they both release their secretions right here at the duodenal papillae. All right. So what secretions are those? Well, we saw the pancreas already once in the endocrine chapter, and that's secretion into the blood. So the pancreas secretes insulin and glucagon into the bloodstream where that circulates. But that is actually only a tiny part of what the pancreas actually does. Primarily, the pancreas is a digestive system organ, and what it produces is two major things. One is a bicarbonate solution that neutralizes the acid from the stomach, and the other is digestive enzymes. So, here we are at the duodenum. If we were to go this way, we'd see that the stomach was right here, right? So the stomach, those parietal cells, produce hydrochloric acid, have a very low pH. So the contents of the stomach that come out into the duodenum, all that acid has to be neutralized. Otherwise, it's going to eat the rest of the small intestine. The small intestine does not have the acid protection aspects that the stomach does. So the way we deal with that acid from the stomach is by neutralizing it with the secretion of the pancreas. So bicarbonate is a base, and it neutralizes the acid. So, if you have H+ from the stomach and you add bicarbonate from the pancreas, what you get is water plus CO2. So we have effectively neutralized that acid. So the pH of the contents of the duodenum goes from about 4, which is what we get from the stomach, all the way up to about 6 or 7 through that neutralization. So very, very important stuff, that bicarbonate solution. And then the other important contribution of the pancreas is digestive enzymes. It is the single largest source in the whole digestive tract of the enzymes that do digestion. In other words, that take large molecules and break them down into smaller pieces. It is also the only part of the digestive tract that has enzymes for everything. The stomach, for example, it only has enzymes for proteins. The small intestine, has enzymes for carbohydrates and proteins, but it's only the pancreas that has enzymes for everything. So it has enzymes for carbohydrates, enzymes for lipids, and enzymes for proteins. So without a pancreas, we don't digest very well. So some example enzymes, an amylase, so pancreatic alpha amylase. An amylase destroys carbohydrates. So it takes big carbohydrates like starches and breaks them down into their component parts, which are simple sugars. So we have that one. A lipase, pancreatic lipase, breaks down lipids. So, as I said last time, fats are hard to digest. So without the pancreas's contribution of this lipase, the fat just comes out the other end, which you find in people that have pancreatic disease. It's called steatorrhea, or fatty stools. And it's because without this pancreatic lipase, the fats never get broken down, so they can't ever get absorbed. So we have amylases and lipases. We have proteolytic enzymes. These are enzymes that break down proteins into smaller chains of amino acids. And then also from the pancreas, we have nucleases. So, typically the food that we eat was once alive, or at least parts of it were. So in addition to proteins, carbohydrates, and fats, much of the food that we eat also contains RNA and DNA, right? Because, you have steak. Steak is made of cells. Cells have DNA in them. So the nucleases are enzymes which break down RNA and DNA into their component parts, which are called nucleotides. And then those nucleotides can be absorbed and reused, and it saves the body from having to make those all the time. So when you think pancreas, think bicarbonate and digestive enzymes, because those are the two principal roles for the pancreas. All right, so because the digestive system is big and complicated, there are all kinds of disorders that can occur from it. And your book likes to just give you some examples. What's wrong with this picture? Well, you're not supposed to see the teeth through the gums, right? So this is periodontal disease, which is a breakdown of the gum tissue that holds the teeth in place. Here's mumps. Remember the parotid gland, sits just in front of the ear. Well, it's the target for mumps, or it's one of the places that mumps targets. So the parotid gland swells up, and you can get, this is an extreme case, but usually it looks like chipmunk cheeks. But you can get these huge things. That isn't what makes mumps dangerous. Why did a vaccine get developed? Because mumps was a leading cause of sterility in males. Because not only does the parotid gland swell up, but the testes is affected that way too. And because the testes has a hard covering, the swelling, instead of getting bigger, it just destroys the tissue on the inside. So the reason we all get our mumps vaccine is because it was bad for fertility, not just for funny faces. Let's see. Gallstones, these are those crystals that can precipitate out of bile. They can be all different kinds of colors. Typically they're sort of smooth and glassy. But the problem is they can end up getting stuck in things. By the end of life, most people have some gallstones in their gallbladder if they still have their gallbladder. All right. Let's see. We won't talk about that one. The pancreas, just like any organ, can get infected or inflamed. When it does, it's a particularly nasty situation because the pancreas has inside it all these digestive enzymes, and the enzymes, because they digest protein, carbohydrates, and fats. Well, what are cells made up of? Proteins, carbohydrates, and fats. So when the pancreas is sick, it often leads to autodigestion. In other words, where the enzymes start digesting or destroying the body's own cells. So literally, a sick pancreas can eat its way through the walls that separate it from the rest of the digestive tract. So pancreatitis is particularly nasty disorder. All right. And then the liver can get sick too. This is an example of cirrhosis, where instead of being nice and smooth, the liver gets these kind of hard nodules all around it and inside it. This can be caused by a number of things, but alcoholism is probably the most common cause of cirrhosis. The problem with this is not just a change in shape, but as the liver gets harder and harder, it gets harder for blood to pass through it. So we end up with a backup of blood that's trying to pass through the liver. So some of the common symptoms of cirrhosis result from the resistance to the flow of blood through the liver. So just a couple examples. All right, so a couple of questions. The pancreas produces which of those things? Oh, wait, I'm not logged in yet. All right, now you should get it. Did you get it? The question? All right, I'm going to try again. You get it? Yeah. Okay. I think everybody's here today. Everybody answered? All right, so nucleases, yes, those break down DNA and RNA. Lipases and amylase, that's fats and carbohydrates, so yes. Peptidases and proteinases, those are protein digesters. So the best answer there would be D, all of the above. And that is unique to the pancreas. There's no other organ that produces enzymes that will destroy everything in the food that we eat. So very valuable. All right, we'll do with this one too. Which of the following is not a component of the hepatic triad found at the edges of a liver lobule? All right, so good. Most of you got that. The one that does not belong is C. The central vein is found in the center of the lobule. Around the edges in that hexagon shape, we have at each corner a bile duct branch, hepatic portal vein branch, and hepatic artery branch. All right. So that was the end of chapter 21. So how about we look at chapter 22 then? All right, so these two chapters, the reason why we're not ending and taking an exam now is because chapter 21 and 22 sort of interlock. You know, in chapter 21, we looked at the digestive tract in terms of its anatomy and its function. But in order to understand the rest of the story, we have to look at how do cells use nutrients and how are those nutrients processed by the digestive tract so that they can be absorbed in a state or a condition that the body can actually use them, right? So that's what this chapter is about. We're going to look at how cells use nutrients, and then we're going to look at the path from ingestion, taking a bite, to absorption across the intestinal epithelium for each of the three big food categories. So the carbohydrates, proteins, and lipids. All right. So first we have to understand, what cellular metabolism is. This word that I know you've all heard before, metabolism, is simply all of the chemical reactions taking place in a thing. We know now that what we think of as life or being alive is the result of cells constantly doing things. Cells are very busy places. It's like, there is never a time when there's nothing happening in a cell. So the chemistry that makes life is happening all the time. So we call that chemistry metabolism. And metabolism can follow a number of different patterns. But an important and easy to recognize one is anabolism and catabolism. All right, so anabolism simply means to build up. So, many of you will have heard of anabolic steroids, right? Those are the ones that athletes get into trouble with sometimes. And that's that same root word, anabolos, or anabolism. And it is to build up. So, we have these building block molecules. So we have simple sugars, amino acids, and fatty acids. Well, if we start building larger molecules with smaller molecules, we are doing anabolism. So we're building things up. So, for example, protein synthesis. We take amino acids, lots of amino acids, and we string them together in a big long chain that we call a protein. So we've gone from small molecules to big molecules. So that's anabolism. Another example would be the formation of a starch. We can take glucose molecules, we can string them all together, and it makes what's called glycogen. So we've gone from small molecule to big molecule. So again, anabolism. So why does the body build things? Why does it build larger molecules? Well, one is for maintenance and repair. Things wear out, in the macroscopic world, and they do in the microscopic world too. So protein has to get replaced. It doesn't have an infinite lifespan. So one of the reasons that cells are building things all the time, making large molecules, is to replace or repair what was there before. So that's one reason. Another is growth. Particularly like in a child, they're physically getting larger. So in order to physically get larger, well, you have to make large molecules to make that happen. You've got or think about a skeletal muscle. You've started a new exercise regime, and your muscles are getting larger. Well, how does that happen? Well, you're turning small molecules into big molecules, into big proteins. So that's another. Secretion, if a cell is making something and then releasing it, well, it's going to have to make that with something. So very often, secretions come from small molecules that have been made into larger molecules. And then finally, to store things. Because of the nature of chemistry and really physics, the more molecules you have, the harder they are to deal with. So if the body's going to store up nutrients or building block molecules, instead of just keeping them as simple molecules, it will string those molecules together. So like that starch, glycogen, for example. The body has a hard time storing 1,000 molecules of glucose, right? But if it can attach all those glucose molecules to each other, it can make one molecule of glycogen, which is much easier to store. So that's another reason for anabolism, is to build up the nutrient reserve. Okay, so anabolism is to build up. Catabolism is the opposite. Catabolism is to break down. So one example of catabolism that I've been talking about for the last few sessions is digestion. Digestion is taking a big molecule, breaking it into smaller pieces. Anytime you go from big to small, that's catabolism. So if anabolism is building all these things, catabolism is the reverse process. So why would we destroy complex molecules? Because let's say that it's not needed anymore. So that's one example. Or it's old and it doesn't work quite right, so we're going to destroy it and we're going to recycle the pieces. Sometimes I think humans think we invented recycling. The living cell has been doing that for eons. Why have to find a thing again when you can just reuse it? So catabolism allows the same building block molecules to be used over and over and over again in different things. So catabolism is the opposite. And both of these processes, anabolism and catabolism both, they work on what we call the nutrient pool. So inside of cells and inside of the body, there is always a pool or a collection of nutrients. And by nutrients, I mean building block molecules. Amino acids, sugars, fatty acids. Those are the nutrients in the nutrient pool. So we can pull things out of the nutrient pool for anabolism, and catabolism puts things back into the nutrient pool. And the nutrient pool is, the whole body works together on that. Because while each cell maintains a pool of nutrients inside itself, the digestive tract is going to replenish those nutrients all the time by bringing in nutrients from the outside. All right, so nutrient pool, anabolism, catabolism. All right, so, and then the other reason we need nutrients is to make energy. So anabolism and catabolism are about, making large molecules or breaking those molecules down. But we also need energy to do those things. Particularly anabolism requires energy. So the nutrients can also be put into the mitochondria, and the mitochondria can create ATP and heat from them. So essentially, the ATP is the mitochondria is like a furnace. You put carbon compounds in, and heat comes out, and as well as ATP, which can be then used for other things. So we're going to see, this is sort of the overview, and we're going to look at some of these steps in more detail. And the first part that we're going to look at is what happens in the mitochondria. Because we've all heard, that the mitochondria produce ATP. Well, yes, they do. But how do they do that? And thankfully, I am not asking you to know all the chemistry steps here. So make a note in your notes that I'm not asking you to learn any chemistry. So like in this slide, for example, there's all of these names and chemical reactions. That is for a biochemistry course, which is not what we're in today. So when things get complicated like this, look off to the side, because usually I'll tell you what I want you to know, okay? Not all of this material. All right, so what happens in the mitochondria? Well, there's two parts to ATP production. One is the citric acid cycle, and the other is oxidative phosphorylation. So inside the mitochondria, we have these two pieces. The TCA, that's this one, the citric acid cycle, and we'll call it OP, oxidative phosphorylation. Okay, so, and the two are connected. They're like gears. One gear turns, and that turns the other gear. All right, so what goes into the mitochondria are short chains of carbon molecules. So if you take glucose, for example, glucose is a 6-carbon sugar. So glucose looks like this. It's a ring like that. Too big for the mitochondria. The mitochondria can only deal with 2 to 3 carbons, carbon sugars, or carbon fragments, rather. Okay. So if this is too big, well, we all know glucose is a primary fuel of the body. So how does this work? Well, we take this 6-carbon sugar and we break it into 2 3-carbon sugars. See, 1 carbon, 2 carbons, 3, 1, 2, 3. Now it's small enough to get fed into the mitochondria. So what we have up here at the top is the carbon fragment comes in. And so this is going to be a 2 or 3 carbon fragment. And then from there, it enters this very complex cycle, which, is not important to memorize. But so the 2 or 3 carbon fragment is added to acetyl-CoA, or acetyl-CoA. Now it forms citric acid. That's how this cycle gets its name. The citric acid is then broken down. So 1 carbon molecule comes off as a CO2, and what's left is now a 5-carbon chain. So we started with a 2-carbon chain. Acetyl-CoA is a 4-carbon chain. So we added our 2 to a 4, we got a 6, and then we see it drops to 5, and then 4, and then 4, and then we add 1 again. All right, so what do I want you to know about this mess? That carbon fragment goes in, and CO2 is produced. You see, here's a CO2, and here's a CO2. Because we know that as humans, we need oxygen, and we create carbon dioxide, right? Well, this is where the carbon dioxide is created. So inside the mitochondria, as carbon fragments, 2 to 3 carbon chain fragments are broken down, CO2 is produced from those carbons. So in the citric acid cycle, we have 2 carbon molecules that go into the cycle, and 2 molecules of CO2 come out. So the carbons have been accounted for, right? So this is the reason we have to exhale, is because the mitochondria break carbons off of these fragments and form CO2 out of them. All right, so that's one part. But that isn't really what this cycle is all about. This cycle is supposed to make ATP somehow. So where do we get that? Well, it's a 2-step process for ATP formation. So we've seen where the CO2 comes off, but more importantly are these steps here. So NAD and FAD are hydrogen carriers. It's the easiest way to think of them. So as this carbon fragment is broken down in this cycle, these NADs are loaded up or energized with hydrogen ions. So we have 2 here, 2 here, and 2 here of NADs. So that's 6. So in one turn of this cycle, we get the production of 2 CO2 molecules, and we get the production of 6 charged up NADs. So a charged up NAD is an NADH. And we get 1 molecule of FAD that gets double charged. So it gets to be FADH2. So it's holding on to 2 hydrogen ions. Why is that important? Because these hydrogen ions are what are going to power the production of ATP in the next step. So all told, what do I want you to know about citric acid cycle? Just to give you a sense of complexity, in a biochemistry class, you would spend probably a month talking about just this cycle. So what I've tried to do here is take a very complicated idea and give you a very simple way to understand it. Okay, so let me know if I'm failing on that. But, so what do I want you to know? 2 carbon fragment goes in, 2 CO2s come out. Why would I want you to know that? Because that's where CO2 comes from that we breathe out, right? And then in that process, we charge up 6 NAD molecules, and we charge 1 FAD molecule with 2 hydrogens. All right. Because then in this step, we're going to use those hydrogens to make ATP. All right. So that previous slide was the citric acid cycle. And what it does is it creates free hydrogen molecules that then are used to power the next step, which is oxidative phosphorylation. All right, so what are we looking at here? So those NADs and FADs had hydrogens added to them. When they get to this cycle, or this part of the cycle, those hydrogens are released. And they pass through, the mitochondria has membrane that sort of reaches down into the inside. So here are the NADH and FADH2 coming in. So first we take the electrons off, and we put them down this sort of hill of proteins called cytochromes. And then we take the hydrogen ions, and we put them into this space. And then on the other end, we have this kind of gate-like protein that as a hydrogen ion passes through that gate, it creates an ATP. So think of it as kind of like a turnstile, one of those spiky things that you kind of have to push and it turns and lets a person in. Well, as the hydrogen ion passes through this protein, it turns a wheel that puts a phosphate onto an ADP. So essentially, it's like a hand crank. One turn of the crank takes an ADP and a P and makes an ATP out of it. So every time a hydrogen ion passes through here, we get the formation of an ATP molecule. All right, so, and then the electrons that were taken off here, they end up attached to an oxygen atom, and 2 of those hydrogens, after passing through the gate, they encounter this oxygen radical, and those 2 things together make water. So in the mitochondria, we have, the sort of overall reaction is that we take a 2-carbon fragment, we put it into the mitochondria, we get CO2 plus water plus ATP out, right? Because we saw the CO2, here's the CO2 that gets made, and here's the water that gets made, and in order to make that water, we have to put oxygen in, right? So why do we breathe in? Because we need oxygen. Why do we need oxygen? Because without oxygen, these hydrogen ions don't have anywhere to go, which means that they don't go through the gate, which means that the ATP is never made. So the reason we need oxygen is for this step right here. It's to grab those hydrogen ions, make water out of them, and in the process create ATP and H2O. So it's the reason why we need oxygen. We have to have that to make this step, this last step work. All right. So a lot of complexity here, but here again, on this slide, my text is what I want you to know, okay? Not all of those steps. Is that something interesting? Sure it is. But I just don't think you guys you don't need to know that detail. Just know about water, oxygen, CO2, and ATP. All right. Okay. So that nutrient pool is not, doesn't function all by itself. Instead, it's in constant sort of communication with the digestive tract, because the goal of homeostasis in terms of nutrients is that every cell of the body will have every nutrient it needs when it needs it, right? Because that's the best thing for health is to have what you need to build when you need to build it. So the nutrient pool is found both inside of cells and outside of cells. And the three building blocks, for sure know those: fatty acids, simple sugars like glucose, and amino acids. Because from this nutrient pool, we can go up, we can do anabolism, which, we form larger molecules, like triglycerides are three fatty acids stuck together. Glycogen is a long chain of glucose molecules. Proteins are long chains of amino acids. So when we go up, we're into anabolism, or we can go down. We can feed those nutrients into the mitochondria by breaking them into 3 or 2 carbon chains. And then those chains can be fed into the citric acid cycle, creating CO2. And then from there, those NAD and FAD, the hydrogen ions, go to the electron transport system, or the oxidative phosphorylation. And as long as there's oxygen, we'll get ATP and water out, right? So this is sort of the overview. We can either anabolize and make bigger, or we can catabolize until all the way down to CO2, right? If you take a carbon or an organic molecule, the smallest thing you can break it into is a single carbon molecule, right? Like CO2. So anabolism, metabolism, nutrient pool. All right. So what contributes, or what are the players in the nutrient pool? Well, one we've been talking about for a week and a half, and that's the digestive tract. The digestive tract is going to break down large molecules into smaller pieces, and then those smaller pieces get absorbed and become part of the body's nutrient pool. The same, you eat a steak dinner, that protein gets broken down into amino acids, and then your cells use the cow's amino acids to build muscle or, protein for you. So it's a nice system that means you don't have to build the whole thing. You can start with a building block. So that's a major contributor. The liver helps to manage the nutrient pool. One of the functions of the liver is to keep the nutrient levels at their appropriate levels. So like the triglycerides and glycogen, the liver has storage of both glucose and fatty acids in that way. So when the body needs glucose, the liver can release it. When the body has excess glucose, the liver sort of gathers that glucose up for storage for later. So that's another spot, the nutrient of the liver. Adipose tissue stores up fatty acids as triglycerides, as we'll see a little later on next time. Fatty acids are great sources of ATP because the mitochondria like to eat 2 carbon fragments, and fatty acids are long chains of carbon molecules. So you can, if you think about, you have a long sandwich, so to speak, and you can break it into smaller pieces. Well, the fatty acids that the adipose tissue contains can create a lot of 2 carbon fragments that the mitochondria then use to make ATP. So more on that a little bit later. But that's another role, fatty acid storage. Skeletal muscle contributes to the nutrient pool because it not only stores some glucose and fatty acid, but most of the protein by weight in your body is found in skeletal muscle. So in the starvation case, where you have had very little protein in your diet for a very long time, skeletal muscle proteins start to break down to keep the rest of the cells of the body alive. So they can play a role in the nutrient pool. And then we have the users. So these are the contributors, the fat cells, the digestive tract, skeletal muscle. But the cells of the body also have to use the nutrients as well. So like nerve tissue can only use glucose as a fuel. So it has to have a constant supply. And then the rest of the cells in the body can absorb and use as fuel all kinds of different molecules, as we'll see as we go. So it's all about this center box here, which is the nutrient pool and its various sources. All right, so we're out of time. We're going to stop there for today.